Bismillah ar-Rahman ar-Rahim. Today we will discuss the reversible and irreversible cell injury and the sequence of events that occur during the cell injury. And this is for third year M.B.B.S students, general pathology. The major targets of cell injury are the cell membranes, the including the cell plasma membrane and the membranes of the organelles like endoplasmic reticulum, lysosomes, Golgi apparatus, mitochondria, and the other target of cell injury are the DNA, the ribosomes, which are the protein forming structures forming the structural proteins and enzymes, and the mitochondria, where oxidative phosphorylation occurs and results in the generation of ATP, which is a type of currency and used by many types of cellular processes. Now, whenever a cell is injured, it will pass through a reversible stage, and then it will go into the irreversible stage or the necrosis. In the previous lecture, we discussed that cell injury during its initial stage will induce cellular adaptation. But if the cells are unable to adapt, or if the adaptive capability of the cells are exceeded, by what we mean that if the injury is persistent or the injurious agent is stronger, so what will happen? Initially, changes will occur at biochemical or molecular level. Now, these changes are followed by loss of function of the cell and loss of function of the whole organ, which is followed by death of the cell. Now, if we want to see the changes that are associated with the cell injury, so initially we can see these changes by using the electron microscope, and the changes that we see by the electron microscope are the ultrastructural changes, and later on we will be able to use the light microscope and we can identify certain changes. So, passing from the adaptation reversible cell injury and then into the irreversible cell injury. Here, what will happen? First of all, in myocardial infarction, when the heart is deprived of its blood supply, initially what happens? That first there is loss of function. This loss of function is then followed by cell death. When cell death occurs, we can see then certain ultrastructural changes using the electron microscope, and it will be later on when we will be able to identify certain changes by using the light microscope. So, in this slide you can see that loss of function occur in first 1 to 2 minutes, right? Then cell death occurs in 20 to 30 minutes, and therefore you know that patients of myocardial infarction, they should be hurried to emergency units of the hospital, because after 20 to 30 minutes then we are left with no treatment or no choice. So this is the reason, and body cells, they differ in their capability to suffer loss of oxygen and loss of blood supply. The most vulnerable cells are the nervous cells, which can hardly tolerate lack of blood supply for a minute or so, and then are the cardiac muscle cells, which can tolerate loss of blood supply for 20 to 30 minutes, and then coming to the skeletal muscle cells and fibroblasts, so they can remain alive for hours. So, for the study of the changes associated with cell injury, we use the electron microscope, and which can detect changes in the cell before we can see changes through the light microscope. So the normal cell when it is stressed, it will pass through the stage of adaptation, and if the cell is unable to adapt, or if the adaptive capability of the cells are exceeded, the cell will become initially reversibly injured. And if the injurious agent is removed, if the cause is removed, potentially this reversibly injured cell can return to normal. But if the injury is persistent, if it is severe, if the injurious agent is stronger, it will cause the cell to pass from the irreversible from the reversible to irreversible stage, and here the cell will die by necrotic cell death. Now, the microscopic changes that we can see through a light microscope, two changes are there. One change is the intracellular accumulation of water, which is also known as the hydropic degeneration or cloudy swelling, and we can see cytoplasmic vacuoles. You know that when the cells are deprived of the oxygen or blood, what happens that the mitochondria, which are the powerhouse of the cell and where ATP is generated by the oxidative phosphorylation or aerobic glycolysis, what will happen that this ATP is needed by the cell for many energy dependent functions. So when there is no ATP, what will happen that the sodium potassium ATPase pump in the cell plasma membrane, which keeps sodium outside the cell and potassium inside the cell. What will happen when this pump is not getting the ATP, it becomes damaged and the sodium rush into the inside the cell, and you know potassium will go out and sodium will pull water after it, and there will be abnormal water accumulation in the cell cytoplasm, and there is swelling in the cell plasma membrane, in the membranes of the ribosomes, in the membranes of the mitochondria and other organelles. So this is the cellular swelling or hydropic degeneration is a change that we can see through light microscope in reversibly injured cells. The second change is the fatty change. But remember that fatty change is most obvious in those cells that deal with fat metabolism. For example, liver hepatocytes, cardiac muscle cells, and to some extent in the kidney and skeletal muscles. Now, certain changes in reversible cell injury that cannot be seen through light microscope and which are known as the ultrastructural changes and for which we will use electron microscope. These changes include the formation of membrane blebs, which are outpouchings in the form of balloons, and the purpose is that to cell becomes swollen, forming blebs so that it can accommodate more and more water. Moreover, there is loss of microvilli. Now, what will happen to the mitochondria? There will be swelling, mitochondrial swelling due to accumulation of water, and here there will be formation of phospholipid-rich amorphous densities, which are formed from the damaged membranes and contain masses, whorled masses of phospholipids. Endoplasmic reticulum, when it becomes swollen, we know that the ribosomes are attached to the rough endoplasmic reticulum. What will happen that the ribosomes, they will be detached from the rough endoplasmic reticulum, polysomes will be converted into monosomes, and as a result there will be decreased structural proteins and functional proteins or enzyme synthesis. The myelin figures, which are derived from the cell membranes and membranes of the organelles, they are seen and they present whorled masses of phospholipids, sometimes with calcium there. And they are known as myelin figures because they appear like the myelin sheath that we know around the nerves. So the two changes, fatty change and cellular swelling, these changes can be seen through the light microscope, and in addition to these changes there will be dilation of the endoplasmic reticulum, and as you know that when the cell is deprived of oxygen for getting ATP, the cell will switch from aerobic respiration or oxidative phosphorylation due to the lack of oxygen into anaerobic respiration or anaerobic glycolysis. So what will happen? The glycogen present in the cell, it will be burned out, and this process will result in the formation of lactic acid, and lactic acid will reduce the pH of the cell. The cell becomes acidic, and the acidic environment will result in clumping of the nuclear chromatin. Additionally, we can see myelin figures, which are voron masses of phospholipids and calcium, and they are known as myelin figures because they resemble the myelin sheets around the nerves. Now, the two changes, hydropic change, it can be seen in the cells in this slide, and it is one of the early signs of cellular degeneration in response to injury, and it refers to accumulation of water inside the cell. And in this slide we can see that in the tubular cells of the kidney, which where hydropic degeneration commonly occurs due to ischemia and decreased production of ATP from the mitochondria. Here we can see on the left side the normal kidney tubules, but on the right side there is ischemic injury. The cells there are deprived of oxygen and oxidative phosphorylation, and here we can see some cells that have become swollen. Now the fatty change. Let us go to briefly discuss the second change. The first change was cellular swelling, hydropic degeneration, and the second change is the fatty change. These two gross changes we can see through light microscope. So before going to discuss the fatty change in the cells, let us go back and remind that free fatty acids, which are derived from the diet and also from when they are mobilized from the adipose tissue, they are taken into the liver, and inside the liver some free fatty acids are also derived from the acetic acid. So what happens to these fatty acids? They are oxidized for energy, converted into ketones, and some are converted into phospholipids, which is a part of the cellular membranes, and some are converted into cholesterol ester. These fatty acids in the liver cells combined with the glycerol, particularly the alpha glycerol phospholipid, and form triglycerides again. Right? Triglycerides are broken down into fatty acids and glycerol, and fatty acids and glycerol unite together to form the triglycerides. Now you know that fats, they are not water soluble, they are lipid soluble. So for to carry them in the blood, they need some sort of vehicle, and this is the apolipoprotein, which combines with the triglycerides to form the lipoproteins like HDL, LDL, VLDL, chylomicrons. So these are the lipoproteins, and this is a way of carrying lipids in the blood. Now what will happen in cell injury, hypoxia and mitochondrial dysfunction? That the triglycerides, they will accumulate in the cells. Here the oxidation of fatty acids, it is decreased. There is increased synthesis and uptake of fatty acids, and particularly the decreased hepatic secretion of VLDL out of the cell. So what will happen? That triglycerides, they will accumulate inside the cells, and this we know as the fatty change or abnormal fat accumulation in the cell. During staining, tissue processing, we deal the slides with xylol, which dissolves the fat content in the fat cells, and the fat cells then appear as empty cells, as we see in this slide. Now when a reversible injury will progress into irreversible injury, two things are very important. When there is an ability to correct mitochondrial dysfunction and to correct the oxidative phosphorylation and ATP generation. Now once the mitochondria had got damaged so much, that even if we restore the original insult or injury, but if they are irreversibly injured, they will get no repair and there will be no ATP generation, and now it is unavoidable that the cell will go from irreversible from reversible to irreversible injury. So one thing is the damage to the mitochondria, and the second thing is damage to the cell plasma membranes and lysosomal membranes, because the hydrolytic enzymes, they are contained within the lysosomes, and when the lysosomal membranes are damaged, they rupture, and the hydrolytic enzymes, proteases, endonucleases, hydrolysis, they are released and they will cause dissolution of the cell organelles. And if the cell plasma membrane is damaged beyond repair, what will happen? That the cell will rupture. So the two important things are the inability to correct mitochondrial dysfunction and irreversible damage to the plasma membrane and membranes of the cell organelles. Now we know that once a reversible cell becomes irreversibly injured, so what we see next? Failure of mitochondrial repair. It will lead to lack of energy and ATP generation. There is failure of plasma membranes and organelle membrane repair. What will happen? The lysosomal membranes, when they are damaged, there will be lysosomal release of lysosomal enzymes in the, for example, hydrolysis, endonucleases, proteases. These enzymes will cause dissolution of the cell organelles and the cell will undergo necrotic cell death. Now, the cell death associated with loss of membrane integrity and cellular dissolution by the lysosomal enzymes. These lysosomal enzymes, they are derived from, number one, from the damaged cells, those cells that undergo necrotic cell death and their lysosomal enzymes are released. And second, the area where cell death and cell damage occurs, here what happens, that the healthy tissue in the immediate environment, it is damaged and it will result in inflammation. Here inflammation occurs, and we know that during inflammation the leukocytes, they are recruited and activated and they will release lysosomal enzymes also additionally to those released from the damaged cells, and collectively they will cause in the dissolution of the injured cells. Now, morphologic changes, we divide what happens in necrosis into cytoplasmic changes and nuclear changes. Cytoplasm, it will appear more pink because during tissue processing we deal the tissue with hematoxylin and eosine stain, HE stain. So what happens, that the eosine stain, it will cause pink cell to appear pink, and the hematoxylin stain, it will stain the nucleus and that will appear blue, and eosine, it will stain the cytoplasm. So what happens in cell injury, that the cytoplasmic proteins are denatured and there is increased binding of the pink eosine to these denatured cytoplasmic proteins, and under the microscope we will see increased pink appearance of the cytoplasm, that we say increased eosinophil. Normally when RNA is present in the cytoplasm, it will be stained by hematoxylin and it will give blue appearance to the cytoplasm. But in cell injury, what happens, that the RNA present is degraded by the RNases, the enzymes that degrade RNA, the endonucleases. So there will be decreased blue appearance of the cytoplasm but increased appearance of the cytoplasm with pink appearance, where eosine appears more prominent and hematoxylin appears less prominent and fat. Now, loss of cellular organelles and loss of glycogen from the intracellular from an intracellular region of the cell will give a glassy homogeneous appearance. Because the glycogen present there, it will be lost, and the cytoplasm will appear vacuolated, which is known as mouth eaten, because the hydrolytic enzymes, they will cause digestion of the organelles which are present in the cytoplasm. And we will also see the oral phospholipid masses which are derived from the cell membranes known as the myelin figures. So these are the changes that we see in the cytoplasm. In the nucleus we see changes, and that are mainly from the result from the breakdown of DNA and chromatin present there. One change known as the karyolysis. The DNase enzyme, it will cause degradation of the DNA, and you know that the DNA in nucleus normally it is stained by hematoxylin and appears blue. So when the in hydrolytic enzymes DNase degrade the DNA, so its blue appearance will fade, and this we say as the karyolysis. As I said that during cell injury there is a switch from aerobic respiration to anaerobic glycolysis, which results in lactic acid accumulation and acidic pH, and this acidic pH will cause shrinking and clumping of the nuclear chromatin, and this is the nucleus will appear in a shrunken solid contracted mass. This is known as the pegnosis. And the third change is known as the karyorexis, in which what happens that the pegnotic nucleus, it becomes fragmented and the fragments fall out. Pegnosis is the nuclear shrinkage which will give increased basopilia and condensation of the DNA into shrunken solid mass. Karyolysis is due to the DNase enzymes which degrade DNA, which is stained by hematoxylin. So automatically the basopilia it will fade, and karyorexis is the pegnotic nucleus when it is fragmented into pieces and go away. In 1 to 2 days, the nucleus in a dead cell completely disappear and we can't see the nucleus. So in this slide you can see the normal cell at the top and the karyolysis with the you can see that the blue color is very faint and there is fading of the basopilia, and pegnosis you can see the nucleus has been shrunken and condensed into a solid mass due to acidic environment that is produced by lactic acid accumulation due to anaerobic glycolysis, and mostly low is the pegnotic cell which is broken down into fragments. So here in this slide we can see normal tubules, kidney tubules, and reversible injury to the renal tubules, and on the third section the renal tubules, they are irreversibly injured. Now myelin, what are myelin figures? Myelin figures, they are derived from the damaged cell membranes, and they present in the form of oral phospholipid masses. Right? Now what will happen, that either they will be phagocytosed by the inflammatory cells, and sometimes they are degraded into fatty acids, and where calcium the salts are deposited and saponification occurs due to calcification, addition of the calcium. Now clinically what we get from a damaged cell and how things help us to detect damage. For example, the cardiac muscle cells, they contain certain proteins in the form of creatine kinase and troponins, troponin I and troponin T, which are routinely detected to detect myocardial infarction in patients suffering from chest pain. So the mechanism behind it is that when the cardiac muscles are damaged, these contain proteins in the form of creatine kinase and troponin I and troponin T. They are released into the circulation and they result in high levels of their blood concentration. So we can say, or we can, if we get high levels of the troponins and creatine, CKMB, that is known in the blood level, we can say that there is some damage to the cardiac myocytes and myocardial, there is myocardial infarction. But remember that these enzymatic tests, they will become positive only 2 hours later after the infarction occurs. So they are not helpful in the immediate diagnosis of myocardial infarction, for which we will use the clinical signs, symptoms and the ECG. Similarly, when we come to the liver bile duct, common bile duct and hepatic duct, cystic ducts, they contain the enzyme alkaline phosphatase. Remember that alkaline phosphatase is also present in the bones. So if we get elevated level of alkaline phosphatase, and particularly if there is concomitant increase in the level of the GGT enzyme, so we can say that this alkaline phosphatase is released from the damage to the hepatic biliary duct system. Similarly, the liver hepatocytes contain the transaminases in the form of alanine transaminase and aspartate transaminase, and the increasing and decreasing level of these enzymes we commonly use in liver diseases, particularly in conditions of liver inflammation in the form of hepatitis. And these transaminases which are contained in the liver cells, when the liver cells are damaged, when they are irreversibly damaged, and when they die, these hormones are released and their concentrations in the blood is increased. Thank you very much.